Okay, I have good news for you. This is the first week you're not going to have to make a smear, stain a slide. You guys get a break from that. I know how much everybody loved that last week. So this week we're going to start doing a series basically from here on out. What's going to happen is we're going to inoculate tubes one week and then look at our results the next week. So the majority of the labs from here on out are going to be 2-day labs. They start one day and finish the next. Some of them will be done in one day when we look at the protozoa and things later, but from here on out, just be prepared to start one week and finish the next, okay? So the first lab that we're gonna do today is going to be the transfer bacteria aseptic technique, starting on page 71. Now, for this particular procedure, we're gonna need both the loop and the needle. Now, we haven't used the needle yet, so it's just like the loop except for it's straight. And you want to kind of make sure you have it as straight as possible. Okay, we don't want too many bendy loops. Okay, also for that, you're going to need several different tubes. So on my rack right here, I actually have 9, I have 12 tubes. Okay, I have 3 media tubes that you're going to put bacteria into, and we call that inoculation. So when you take a bacteria from one tube and put it into the other, it's called an inoculum, and you are inoculating those tubes with the culture, okay? So these 6 tubes right here are the tubes that I'm going to be putting my bacteria into or inoculating, and then these 3 tubes here would represent the bacteria that we are going to put into the tube. Now today I'm just going to pretend I don't actually have any bacteria here. I'm not going to run the flame, wear goggles or anything like that. However, you know that when you're in class, if there's a flame going, if you have your bacteria that you're using, you're going to be wearing goggles and gloves. Okay, so first thing that we're going to do is look on procedure 7 on page 72. There's a picture, and you can see it has the L. lactis, which is the bacterial Lactococcus lactis. Then the other bacteria is Pseudomonas aeruginosa, and the third bacteria is Proteus vulgaris. Okay, what you're going to do is you're going to take each bacteria and put them into the 3 separate types of tubes. Okay, so the 3 separate tubes I have here is I have broth tubes, so those are the liquid. Ooh, don't do that, don't tip it too far. Then we have the slant, which we've seen before, it's a solid, okay? And then we have what are called semi-solid agar deeps. That means they're solid, but they're not have as much agar in them as these do, so they have a little bit of pliability, okay? Now in your picture, it shows that you're putting the lactolactis into the 3 separate tubes. The broth, the slant, and the deep, okay? And then you're going to put the other 2 bacteria into each of the 3 tubes. So what I would recommend you do first, and which is always recommended when transferring bacteria from one tube to another, is you label your tubes first, okay? So you'd use your Sharpie that you have, and you're gonna write the name of the bacteria that you're putting in there, and some sort of a name to identify your group, okay, or your name or something like that. And you can also put the date on there if you'd like. Now, when we do this, you're going to be working in groups of no more than 3 people, okay? And I want every person in the group to be doing their own inoculation. What that means is I don't want each person to have a separate set of tubes. I want each group to have the 12 tubes, the 9 that you're putting into, and the 3 bacteria, but I want each person doing a transfer, okay? So maybe you could say person A is responsible for transferring lactolactis into these 3 tubes, person B is transferring the Pseudomonas, and person B is transferring the Proteus vulgaris. I do not want to see one student holding the tube of broth and another person holding the culture, and one person dipping from the culture and the other people working as a team that way. I want you guys to work as a team to help each other understand, but I want every single person doing their own inoculations, okay? So we have all the tubes labeled. I'm going to pretend they're all labeled and they're ready to go, okay? So now we're going to use good sterile technique, which I kind of taught you the first week of class of how I don't want you to ever set anything down. So of course my flame would be going, okay? And we're going to be using the needle and the loop. So what I'd like to do is start with the broth. Okay, oh, I have these all backwards. I should have it this way, where I have a broth, a slant, and a deep. a broth and a slant and deep for each bacteria, okay? So I'm going to pretend that this bacteria here is the lactolactis. So I'm doing the first one, okay? And remember I told you to use all your hands and all your fingers, okay? So I'm going to go into the broth first. Now, the first thing that you want to do, of course, is to sterilize your loop, okay? And then, of course, you're going to let it cool off. Now, this one would be the culture, and it would be labeled with the culture's name. That's what you're taking from. And this one would be the broth that I'm putting it into. Now, remember, if it falls to the bottom, kind of want to do the tapping to bring it up, okay? So you're going to sterilize your loop, and then it's going to cool, like we've done before. Now, I want you to hold both tubes in one hand, okay? I don't want one tube here, and this all kind of, I want you to get used to holding 2 tubes in one hand, okay? After I've sterilized my loop and I'm ready to transfer from here to here, I'm going to use my fingers again, okay? This time I'm using 2 fingers. I pulled off both of them, flame both of them, and then I'm going to dip into the culture and then go directly into the broth, and you can kind of shake it a little bit. Then I would flame my tubes again, put their lids back on, and flame my loop. Okay, now that's the one I just finished. All right, so I'm flaming my loop, it's cooling off. Once again, I'm going to pick up my culture. Then I'm going to pick up the slant next, because you're still going to use the loop for the slant. Okay, so then I have my culture and my slant that I'm putting into, and a fly that's bugging me. I've already sterilized my loop, so now once again, I'm going to pull them off with my fingers. Never ever set your lids down. I'm going to flame them. I'm going to go into my broth, and then to put it on the slant, you're going to start at the bottom and just gently run it along the slant. And you flame them, and then you close it and flame your loop. That fly is bugging me. All right, so when I put it on the slant, you never gouge the auger, you just run it along there from the bottom up to the top, okay? Now the third one is going to be the deep. And for the deep, you're going to use your needle. So we flamed this, we've sterilized it, now we're going to use our needle. So the same thing, we have our culture that we're taking from. Now this is the semi-solid auger deep. At this point, we're actually going to stab it into there. So I've already have sterilized my needle, okay? I'm going to take off my 2 lids, run them through the flame, go into my culture, and this time I'm going to, like I said, we're going to stab it. You're actually going to push it down in one motion, in and out. Okay? So of course the straighter your needle is, the better it's going to be. So I've sterilized that. Here's my original culture. Here's the stab I put it into. So like I said, you just go straight down into it and straight out. So then you do that exact same procedure with the other 2 bacteria. And like I said, if you have one person assigned to this bacteria, another person assigned to this one, and another person assigned to this one. And I want you to put both tubes in one hand. It's going to seem a little bit awkward at first, but it's the way that I'd like for you to learn to do it. Then you're going to take all your tubes when you're done with everything, and you're going to put them in the incubator. So make sure they're labeled. We're going to have to condense racks between different groups so that they'll fit in the incubator, so you may want to like identify your group with some sort of a symbol or something so that it doesn't get mixed up with other groups. Okay, that's aseptic technique.